Good evening. Today we are going to learn about the chapter 8, which contains the enzymes, which are a biological catalyst. So, what are the enzymes? In this chapter, we are going to learn about what are the enzymes and how it works as a biological catalyst. The diversity of enzyme functions. So, what are the different functions it can show? And how the chemical reactions affected by these enzymes, like whether it is the rate or the functional rate or the reaction rate, and also how these reactions can be performed the faster without affecting the enzymes, and how these enzymes act as a catalyst. So we'll know a couple of enzymes functions examples. So along with these enzymes, we do have some of those like coenzymes or the vitamins, and also some of the essential metals are also part of these catalytic functions, and we look into the various characteristics of this enzyme catalyst. So then also we can learn about what are the inhibitional functions of the enzyme. So like, can we inhibit the enzyme function, or we can regulate its activity? Along with this, we'll also learn about, is there any modifications we can perform on those enzymes to modify its functionality. So there are also some non-protein biocatalysts, like catalytic nucleic acids are there, and we'll learn about various tools which used in the enzyme functionality and determination. Okay, so enzymes as a biological catalyst. So what is a catalyst means? A catalyst is a substance that increases the rate or velocity of the chemical reaction without itself being changed in the overall process. Means if something is reacted or a chemical reaction is happening, like substrate to the products. So when the substrate is utilized and creating into the products, and something drives it towards to ease the function, or it can make it faster so that we can perform this reaction in a short period of the time, or without utilizing a lot of energy. So that is what is the catalytic is known as. So today, this we are going to look into the diversity of enzyme functions. So what are the different functions we do see, and what are the variations on the enzyme function? So in this, we do see several type of enzymes are present. So when we have these different kind of enzymes are present, so it does have various functions. So each substrate has its own function, and each substrate makes into the various products. So based on the substrate, based on the products, so we do have the differences in the enzyme functions. So these are the basic class, like you have the type of food, what they have it, and what the products they have to make it. So we do have several type of enzymes, and those are there. So when we use these enzymes, we are talking about these as a catalyst. So they do change the chemical reactions rate. So whether it can be processed faster, slower, or it can be the temperature uses, or utilizing various substrates and other things. So it depends on various other things. So when we talk about the reaction, so if it is in usually when a substrate is used and made a product, so you can imagine it as a irreversible reaction. Means when it is transforming from A to the B, then it is not going back into A again. So in that case, the reaction rate means how fast it is going to happen. That depends on the rate constant with the first reaction order. So how many reaction orders are happening, one or two or three, and how long it takes time. So unit of velocity and those with the first order of the reaction, so how many time it takes, so that is depends on those. So we can use this formula to identify what is the reaction rate it's going to be. And in that, the order of reaction, it can be also be determined by comparing the kinetic data. So in this, kinetic data means how it is fast it is doing. If it is a first order reaction, so n is equal to 1, so only one time it is happening. If it is a repeatedly is happening, then it is a 2 reaction. Then the n becomes 2. So we can use these formulas to identify the rate of reaction on those. And in that case, we do get the logarithm of the products and the byproducts. So that's what we do have to react. And when determining the order of this reaction, so the graph, this graph, it shows that one, the conversion of A to the how long it takes time. So it does takes faster, or it can go into a linear uh, also. And for example, we have a myoglobin. It's bind into the oxygen and it becomes oxidized. So how long it takes, the myoglobin is bind to the reaction oxygen, and then it becomes myoglobin with oxygen and it releases into. So how long it takes, that can be used, and the reaction, the myoglobins can be determined the constant rate constant. And the rate of chemical reaction also depends on couple of things. So as we mentioned, it depends on the order of the reaction, whether it is doing direct order or it is giving any byproducts. So it depends on that, and also it depends on the concentration of the reactants. So how much concentration we are taking initially? Shall we taking the higher concentration? And also there are some enzymes, it depends on the concentration levels. Like if you are more concentration, they may have feedback uh, uh, mechanism also. So whatever the producing is based on that amount, and is amount, like you think about the glucose. When we have a blood glucose amount, so the insulin production happens, and the moment the blood glucose level goes off, then we stopped the insulin production. But in case if we have long period of the time the glucose levels are there, then we do have the insulin levels are continuously producing, and that's what is important. So it depends on how much is the concentration of the reactants are there, and what is the temperature, and also whether the reactants, where all these are the enzymes, so it depends on the temperature conditions which is favorable. And like you imagine, when you have the lower temperature like refrigerators, so what happens, we do have the refrigerators lower temperature, so there the enzyme activity is slow down. So that's what we need to modify, so that is the reason we do use the refrigerator. And also the value of the rate constant, how much it is fast it is going, or how much it is, so that also it depends on that. So also whether a chemical reaction is also depends on the free energy difference. So how much energy it is going to consume to perform the reaction, or how much it is going to release it. So which one is important? So which takes consume the energy, so it requires a lot of energy to start the reaction, or the reaction which produces the energy. So mostly the energy producers are the favorable ones. And also this transition state and the reaction rate also determines the rate of chemical reaction. It is not only in the initial state but also the final stage. So it is whether it is going to form, or what, whether it is a favorable condition or the situation it is going to be favored. So it depends not only on the initial stage but also it depends on the products. And the transition state is an additional energy barrier to convert the reactants into products. So when this transition state is happening, so when the energy, it takes a lot of energy to heat up or do the things, so that's what it makes it on this one. And it requires free energy to make the reactants or the products, and so you can list stable state during the progress of the chemical reaction it is happening. So look at this diagram, so where you can have these products and it requires some energy to more energy consumption, and then the energy is released, or it is less is there, so that is going through, it passes to converted into the molecule B, then it requires additional energy, the energy transition state it is happening. But with uh, the enzyme, so if we can reduce conversion of the pyranol sugar from the boat to transformation, so we do have the energy level is reduced, like it is consume less energy, so the reduced energy can again transform into the difference between these two energies released and what consumed initial one also depends on the rate constant, the transition rate. The rate constant of the chemical reaction can also increase by raising the temperature of the reaction. So sometimes you know that one, like when you warm up, then the reactions happens faster, because also the free energy availability and those. So it is very important whether, sorry, whether you are trying to move the faster reaction, so you can give some energy to the thing and make the things faster. So this is called the activation energy. You can increase the activation energy. So when you have a uh, temperature raises, so in the low temperatures it requires a lot of high free energy need to be there so that they can initiate the reaction. But in case when you increase the temperature, then in that temperature, so you have things are moving faster and you require less energy to be used over utilized over there, and that is uh, so that is what the raising temperature also increases the rate constant of the enzyme reactions. And the rate constant also of the chemical reaction also depends on the absolute temperature. So the temperature, absolute temperature means the whole temperature differences and everything. And also it is the activation energy. So it has depends on those how much energy is initially you are giving it. So, so enzymes also they have the transition state, like from one to the another products. So based on those products, so they do have the transition. So that is very important to know about how the transition is happening. So for example, if you see these products, this substrates are there, then the enzyme binds to that and it combines, and then the product is made. And when this product is made, they have the transition state from one to the another products are made it, and this transition state, and it may have the one product like direct product to the substrate to the product, or they do have the transition states like having intermediate products. So it depends on that how these products are making. They are the CVP office. So when we look into how enzymes act as a catalyst, so we are talking about like it doesn't involve, the enzyme doesn't involve any of the reaction, but they do act to reduce or modify or the reaction rate of reaction. So how those are working as a catalyst? So if you look onto this, they do work as a lock and key or induced fit model. So in the lock and key, so they exactly, you know how the lock and the key was. They had to be exact same size, the shape, and it has to fit. So in that case, they do the enzyme. The yellow color is the enzyme, or this red color, red color. So they do have the, when the substrate is there, they exactly fit with this active sites of the substrate, and then it transform into the products, and then it is the enzyme is released back. So we don't see any modifications are happening in the enzyme, but at the same time enzyme is utilizing and helps the substrate to be making into the products. So when you look closely, these are the 3D dimensional structures. So the tertiary structure and the quaternary structures of the enzymes. When you look, so they do have the binding site. So that is very important, where this substrate is going to bind to those enzymes. They do have this binding site, and this binding at the binding site, the bind, for example, the insulin. So the cleft, so the glucose binds over there, and then it can induce. And sometimes what happens is to have the stringency of the enzyme specific to, and each enzyme was very specific to the substrate. So and some undergo structural modifications once they have bind it. So like we saw in the myoglobin, so once the it binds with the oxygen, they do undergo with the structural modifications or any of those things. So here we can see the when the glucose is bound, and then the structural modifications happens in those, so that it can perfectly fit with that specific to the substrate. And that is the reason sometimes you might have seen like they do have a competition of the substrate. So that is one type of, like we will know further about how the mechanism it was, like inhibition or other things. So during that time we'll know how we can mimic the substrate structure, so that we can inhibit the function of the enzyme. So this acceleration, when you look into, so when you have the enzyme, so for each it requires a lot of transition energy is required, the transition state. But when you use these enzymes, so in the presence of enzyme, this transition state is reduced, and you can achieve the functionality is in a short with that short energy level, or the transition state is going to be reduced. So to enzyme catalyze, they proceed by one or more of those, like either general acid-based catalyst, they do have a covalent catalysis, electrostatic stabilizations, then they do have the proximity effects. Some they have the preferential stabilization of the transition state. So the transition state is that they have the stabilization, or they do have the conformational change that also play an important role in the catalyst. So in this serine protease catalytic triad, so you can see it forms a these are the various amino acids present on that one and the location. So it does have some conformational changes and other things. They do have it. And in case of the peptide bond hydrolysis, so you can see it forms the utilizing the water molecules. They can react and also modify the shape, and then they do have the this is the example of the chymotrypsin, how it is going to be. And we along with those, we do have some of those coenzymes. So the coenzymes is like they help along with the enzymes. So those are not major enzymes. They don't take part in any of the reaction, but they help for the enzymes. And also the vitamins and some of those essential metals also play an important role in enzymatic actions. So here this table shows some of the coenzymes. In each enzyme it is that one. So these enzymes, these are the either the small molecules or some ions. They bound to the enzyme, and these are not irreversible. Okay, imagine we are not involved in any of the reactions. So whenever we are talking about the enzymes, they are repeatedly utilized, they are used all the time, so they are not going to perish or immediately after that one. So imagine when you eat a lot of food, okay, so you don't need all the insulin is going to produce continuously. It produces and then it works on once the reaction it is happening, and then again the other glucose molecule binds slowly, the glucose is released, and that longs the insulin is circulating in the body. So these are not going to destroy immediately, or so that's the reason the coenzymes also they don't bind irreversibly with the enzymes. And they do have metal ions. So many of those enzymes they do have different metals. So that's the reason one time we can see uh, uh, these metals are also important for us. So when you are consuming, like some people take multivitamins or these metals, these are the rare metals what we don't get sometime with the food, or sometimes we do need a supplement to be taken. So these also play a major role in the enzyme functionality. Then when we look into the kinetics of this enzymatic catalase, so what we say is the initial rate of enzyme. So it can has a particular reaction time and kinetics, everything. So from the substrate to the intermediate products, and then the final products happen. So it has undergo for each one sometime like one substrate to the one product it happens, or if you have any intermediate products, so we may need to use different enzymes for each substrate what we are getting it. So when you have a reaction from the substrate to the product and it has involved with a couple of intermediate products, so here you are expecting to have more number of enzymes involved in that one. So it is not like only the same enzyme is going to work from the substrate to the product. So that time we do have the rate of reactions, and the kinetics also going to change based on the availability of the other enzymes and where it is going to stop. So in total, so we have several type of reactions it's happening. So these are different based on the substrate and based on those uh, other enzymes present, so they do have different uh, rate of reactions and everything. So in this, it depends on again the substrates, various substrates, how those are strongly associated or dissociating, and also it depends on the availability again on the so like when we the best example what you can take it is if you need any of the enzymes have the optimal temperature to be, and you are taking like a glucose is the direct energy source. So the main source of the energy is the glucose, that is the initial one. But the food whatever you take it, like either protein, fats or carbohydrate, the for energy production everything need to be transferred into glucose. So the fats they can have intermediate products, and those finally for energy release they do need to convert into the glucose. So when you have a storage, so what happens, the fat can store if you don't need any energy. Okay, they don't need to transform into glucose. So the other enzymes whatever needed, they are not going to express, so the rate of the whole process is going to change according to the byproducts whatever it is going to use it. So in the total energy the enzyme from the substrate to the uh, product is making, it depends the whole energy transition, but each has its own enzymes, how many it is used and how much free enzymes are released and how much is in the complex it is there. So we can use the Michaelis-Menten equation to see how much total enzymes are present and how much is utilized in each complex. So that's what we can calculate based on these different formulas. So we can here in this class you are not going to right now calculate any of those exactly how much enzyme is used and how much is this, but it is helpful for if you are doing any like further education like masters or graduation the time, definitely you have to calculate how much, so that is going to be useful. And here we can look for the kinetics of the difference, how much and what exactly the substrate, the enzyme is required for making into the substrate into the completely dissociation and those things. Okay. So we can see the maximum achievement of this, the rate, how fast we can achieve, and other things we can also calculate based on those the reactions by line Weaver or Burke plot. So the Michaelis constant Km, it indicates the substrate concentration at which reaction rate is of the velocity, like the reaction rate is happening. So these enzyme, it shows the enzyme activity when the substrate concentration is above the constant, and what is going to be the turnout number, and how much catalysis is required, and what is the uh, enzyme product it is going to make, everything it is, we can calculate accordingly. So when we talk about the specificity of enzymes with the substrate, so when many enzymes they catalyze the chemical reaction, they convert more than one substrate into more than one product. So in that case, these the binding of those substrate, the enzymes to the substrate and the releasing and everything. So it depends on several factors like the random substrate binding or order of the substrate, and also when they have the displacement of the substrate. So in one enzyme is bound and then afterwards it is displaced and another enzyme is bound. So it depends on several factors, and those are called the random substrate binding, and multi-substrate reactions are happening on those. So in this multi-substrate one, so one substrate must bind first before the other can bind. So this is what the reaction it happens, and then various cofactors also coenzymes binds so that it can initiate or it can process faster. So in that multi ping pong double displacement, you can see the one substrate bound and reacts, and they release the first product, and then the second substrate is bound to the reacts and releasing the second product. So this is happens in the example of the chymotrypsin or some of those reactions. It happens in that. And here we're going to talk about some of those enzyme inhibitions. How can you stop the function? Why do you need it function? So sometimes you don't need like in the case of cancer. So you have several different kind of enzymes are activated which you want to stop them too. So that is what it is. Or insulin is continuously circulating. So you want to stop that one. So that is what it is. So many drugs process like uh, whether is anybody is experienced on the Helicobacter pylori. So this causes a gastric cancer also. So it causes very commonly seen bacterial infection, and it causes the ulcers. So the doctor they prescribe you a proton pump inhibitor. So those are the enzymes like when they can transport the protons from one to the another. So when this can be inhibited. So like that. So the development of such drugs has been advanced by kinetic studies, so which based on the structural studies of the target enzyme. So that's what we talked like they have like each enzyme its own structure, and based on the substrate it binds it. So if you can mimic and add a drug which is similar to the substrate, so what happens instead of the substrate, if you had more amount of the drug or the competitive one, so that can go and bind, and then the enzyme is not available for the real substrate, and then in that case it can be removed from the blood or something, or it is no longer able. So that's what it happens. And some natural or synthetic toxins then react also, they involved with the enzyme inhibition or inactivation. So there are like two types of enzyme inhibitors are there. Some of them are like reversible inhibitors, so they non-covalently bond, so they are not easy to detach. And then other is irreversible inhibitor, so those covalently bond to these things. So when we have these drugs are so the reversible inhibition, so it can be reversible, it can be modified again back. So in a reversible inhibition, the inhibitor interact with the enzyme non-covalently, and in an irreversible reaction, the inhibitor is bound covalently. And here we got a competitive inhibition where we talked to have the similar structure of those substrates so that we have the competition so that we can reduce, or uncompetitive, so where we don't need any competition, so we have remove, like we can provide other substrate, or we can do the byproducts, we can stop other, or we can have a mixed one. So in the competitive, we talk like instead of this, we can have a structural similarity of those, and then we can make the enzyme so that it cannot be functional for those. So this is what the competitive one. So in the competition, we do modify those rate of association, and also the reaction maybe sometimes it's faster, or it can be we can slow down so that the enzyme cannot release easily from that competitive one. And in the uncompetitive, what happens is you have the substrate. So we talked about the structural modification. So when this enzyme is binding to, and if it is binds to some other, so it can modify the structure so that it can no longer able to bind with the substrate. So that is what we can do. So non-competitive, that one also we can see how the modifications can be happen on those. And in the non-uncompetitive, so by substrate reaction, in order substrate binds involving the inhibitor, and then that is uncompetitive with the respect to the substrate. So then these modifications happens, and then you can replace with the different structure substrate. Those are in the secondary, or so that's what it cannot no longer bind with the substrate, and it cannot be this one. And sometimes we do have like structural similarities are modifying those structures, so it cannot bind with those things. And in the midst or non-competitive, the inhibitor binds to the site of the enzyme in a different place, and then the substrate, so it can diminish the function of the enzyme in that. And in case of irreversible, so that means once it bind, modify this enzyme, that's it. You cannot take it back. So these are this diisopropyl fluorophosphate. So these are called DFE. It binds to the active site of the serine of acetylcholine transferase, and which is involved in the nerve conduction, and this inhibition enzyme causes the rapid paralysis. So these are the irreversible actions it happens. So we cannot, once the paralysis, it is like in that way. Then how do we regulate the enzyme activity? So we can regulate the enzyme activity by controlling the enzyme function in the cellular at the cellular level. So there are several ways to control or regulate the enzyme activity within the cell. So for example, the reaction rate increases with the increasing substrate. Then we have the large change in the substrate. It would require to change the activity of enzyme. Then, so that's what we can regulate at the cellular level. And it can have the feedback in the inhibition or activation at the control points. So what is the feedback inhibition is once you like you are eating and you say that okay, I ate enough, I don't want to eat. So that is the feedback. Although you see the food, you said I ate enough, I don't want to eat anymore. So like that. So or the activation. So it is not going to activate. And also these are mediated by the allosteric or similar type of enzymes, which is going to be work, or it can also it can maintain the homeostasis of the drug. And in case of covalent, so covalent is a reversible but irreversible, mostly it happens. But in the irreversible, so Melly said signaling processes happen, so they have to process regularly, so that's what it is. Or irreversible, like the blood clotting happens, or the zymogen activation, so that's what those are the covalent modifications that happens, and those are irreversible. And in the feedback inhibition, so when the enzyme, the substrate is there, and enzyme it gives the product E, and once the product E is made, and then it stops the enzyme 1, okay, I'm good enough, we made this final product, so we don't need any more of those. So then it stops the enzyme production and other things. And in the allosteric, so those are the multi-subunit proteins that change their conformation upon binding to the substrate, or they have the epitope. So when they have a homoallosteric, means they have like the so we have this enzyme. So homoallosteric means enzyme which will cooperatively in the substrate binding, and in case of heteroallelic, so these are the non-substrate. So these are enzymes which are regulated by the non-substrate effector molecules. So in the homo allosteric, you can see non-cooperative and the cooperative enzymes. So those can more increase, and the non-cooperative can reduce over the time. But in case of hetero, the non-competitive substrates can also involve, and then they do regulate the inhibition. So many enzymes they covalently modified at the activate deactivate them. So in that case, the covalent modifications are the functional on or off switches it makes it. So in case of phosphorylation, when they are making the phosphorus, so they are carried out by the protein kinase. They can be reversed by dephosphorylation. So they are taking out the phosphorus. Or the zymogens, they convert the enzymes by the inverse irreversibly as a at the proteolytic cleavage site. So those are the enzymes. It can be reversible or irreversible. And reversal phosphorylation and dephosphorylation. So they do change the kinetics and also the phosphatase activity. So whether it is phosphorylation, the phosphatases, which is in presence of the water, it can convert into this, or they do have those enzyme. They do have this different phosphorylation. It happens so from like ATP to ADP, and then they do have this regulation of these kinases. And the zymogen, they do have the proteolytic cleavage. So that's what it comes into different byproducts, and that can also what are the each uh, intermediate substrate can also influence on the other side pathways. So it is very much very commonly seen when we are looking into like in the cell signaling. So when we are having so the one of the byproducts, the other enzymes can the byproducts can influence on the other. That's what the whole signaling pathway is involved, and that is what it whole regulates, and that is the reason, that is this is the one of the major reason of the cancer is not treatable, because these byproducts they can influence on other enzyme pathways, and it is very difficult to know whether we can regulate it or not. Or that's the reason, if we are blocking one pathway, then it goes into another pathway, and then it is regulated again. So that's the reason we don't have a single drug or a single the effective drugs against the cancer. So in the irreversible zymogen activation, so they do have the proteolytic cleavage. So it happens, they do have the different byproducts, and then these byproducts influences on those. And we do have some non-protein bicatalysis. So these catalytic nucleic acids, so the ribonucleic acid, they can catalyze the chemical reaction. So these are ribo RNA molecules. They can also color the ribozymes. So these ribozymes, they had RNA-based self-replication and catalytic mechanisms. And before DNA, so like it's a chicken and egg. Initially, they had it about whether the DNA came first or the RNA. But when you think what is the functionality, so when the DNA is there, DNA transform into the uh, RNA, then RNA translate into the protein. So RNA can be directly trans pass the information as protein. So when you're talking about, so it might be RNA is the first one to coming up in the world so that the RNA can make it into the proteins. But when it comes into the stabilization, so that's what RNA deoxyribonucleic so that deoxy uh, became, so that's what those are more stabilized. So because of the stability, the DNA is formed. So to identify these many of these chemical reactions and other things, so these can be measured under the very steady. So these modifications, so especially the substrates what we are going to generate, we can easily identify those by several reactions. So these can be uh, carried out using like spectrophotometrically. So these substrates, the measurement of those products can be measured, or using the fluorescence. So the reading, the light passes through uh, the buffer, and then the substrate made into the products, so it makes into the different light to be absorbed, and those can be read by the machine. And also we do have the radioactivity assay, so we do have some also like by providing like phosphor or sulfur, which is in the part of the amino acids, and then those are converted, they can be measured, the lighting whatever it is produced. And also these can be devised like these are the main electronic devices. These can steady or get the light, they can, that's what it is, they can react and then see. And also they have the temperature modifications, like whenever you have that whenever the reactions happen, we do have the some of these temperature modification, and those can be also measured. And these everything can be measured as a kinetics, how much it is changing and what exactly it is changing. So that's what we can look into this. And we do have like the converse and the metal ions for their diverse functions. So we do have utilization of these metals and other things. We can still measure of those things. And that's what it is. So this is about various enzymes and its activity, and how we regulate those things. So let me know if you have any doubts and anything. Okay, thanks.